Hello, listeners, and welcome to the DrGPCR podcast, where we bring you conversations with leading experts in GPCR biology. In each episode, we dive into the career trajectories, discoveries, and groundbreaking research shaping the field. The podcast was born at the height of the pandemic with three key goals: to share the latest discoveries, give researchers a platform to showcase their work, and inspire the next generation of scientists to explore this fascinating field. Since our launch, we've recorded over 160 episodes featuring incredible guests like Dr. Brian Roth, Dr. Robert Lefkowitz, Dr. Fiona Marshall, and many more. Our mission is to unite the global GPCR community, encouraging collaboration to improve human health through a deeper understanding of GPCR biology. Thanks for joining us in this exciting journey, and now let's dive into this episode. In case you haven't heard, a DrGPCR University course is coming soon. Let's take a minute to explore what it's all about.
We are excited to have Dr. Terry Kinnakin back at the DrGPCR University. This is a long format course. Terry is here with me this morning to talk to us a little bit more about what to expect in March of 2025. Terry.
Thank you. Hi, folks. This is a 4-lecture course on development of PK, ADME, and safety, which is very, very, very important in drug discovery and development. If you have a compound that is active, useful, but you can't get it to where it ought to be in humans and not causing harm, then it's useless. So this is a critical step. I've been in programs in industry where we've had active scaffolds, but we could not pursue them because we weren't able to develop them properly. This has changed, and so part of this course is to try to introduce you to the advances made in this field. We basically have no excuse to lose a compound because of PK now.
Want to hear more? Go to ecosystem.drgpcr.com/GPCR-courses and enjoy a 25% discount on your registration with your premium membership.
Hello, everyone. This is Yamina from DrGPCR. I'm very excited this morning, very early this morning. It's noon where you are, Brian, to have with me Dr. Brian Hudson. Brian and I met a very long time ago at a GPCR retreat. I think we were both either master's or PhD students at the time, and it's really great pleasure to have you with me today. Welcome, Brian.
Thanks. It's great to be here.
Very excited to have you on. So let's start at the beginning. If you could introduce yourself, tell us who you are and what is it that you do.
So I'm Brian Hudson. I'm a lecturer in the School of Molecular Biosciences at the University of Glasgow. We're part of the Center for Translational Pharmacology that is sort of a GPCR-focused research group based here in Glasgow. I've worked for GPCRs for around 20 years now, recently really focusing on metabolite sensing GPCRs, and in particular developing new tools and approaches to study this group of receptors.
Thank you. And, people might be wondering, how come we met such a long time ago at GPCR retreats? Obviously, that was in North America. So what's your story as a scientist? How did you end up in science, but also how did you end up working on GPCRs?
So, I started as a scientist during my undergraduate degree. I was at a small university in Canada, so I'm from Halifax, Nova Scotia. I went to a small, not particularly science-focused university and started working in a summer research lab. And at the time, I was interested in medicine, and the most medical-like research that was going on at the university happened to be fungal toxicology. So we were studying how a toxic fungus that grows in water-damaged buildings would affect mouse lungs. And it was interesting work, and I didn't become a mycologist. I didn't really follow through on the fungus, but I really got excited about the research side of things from that experience, and that led me into a PhD. And I sort of fell into my PhD position. I'd like to say that I strategically planned my research career, but sort of as it's gone on, I've kind of fallen into the different areas that I've ended up in. So I ended up doing a PhD at Dalhousie University, which is a slightly bigger, more research-focused university, also in Halifax, Nova Scotia. working with a woman, Melanie Kelly was her name, and she ran a research group that was focused on ocular pharmacology. And in particular, she was interested in whether cannabinoids could be used in the treatment of glaucoma. And I joined her research group, and as originally a master's student, and then transitioned to a PhD student before I finished the master's. And when I started, she sort of said, "Well, I work on generally cannabinoids, and what are you interested in?" And she handed me a grant, and I thought, the GPCR stuff was the most interesting bit, and I ended up sort of falling into a GPCR dimerization project, sort of when dimers were, the big hot topic at the time. So I did a project that was originally looking at homodimerization of the cannabinoid CB1 receptor, and later transitioned into a bit of heterodimerization of the cannabinoid receptor with the beta-2 adrenoceptor, and looking at how that might influence particularly the regulation of pressure in the eye. And it's sort of, that's sort of where we would have met, was when I was doing that PhD at the time. I was, being from Halifax, we were a little bit removed from the sort of traditional GPCR retreat community, which is kind of centered around the Great Lakes area. But it was the closest meeting for me to go to, so I sort of went to that. And I think going to that GPCR retreat meeting was really what made me more of a true GPCR person, because sort of the rest of my lab was more of an ocular sort of pharmacology. And then, as I went through and towards the end of my PhD, I was starting looking for postdocs, and again, sort of fell into a chance meeting with Graham Milligan, who had come to give a seminar. And by a very odd coincidence, the week after he came to Dalhousie to give a seminar, I was actually going to Scotland for a cannabinoid conference, and happened to have a few days in Glasgow to spend. I went to Graham's lab and gave a seminar for his lab group as a PhD student, and then sort of a year and a half later, when I was then looking for a postdoc, I got back in contact, and there happened to be a position, and I moved to Glasgow and have unexpectedly been there ever since. Originally, I didn't plan to be here quite so long, but now it's been almost 14 and a half years now I've been in Glasgow, which is hard to believe.
And you're right. Now you're mentioning the cannabinoid story and that you've been doing. That was the GPCR retreat where you were presenting a poster, and I remember we were sitting together with with Terry's lab, and I think that's where we met, and the cannabinoid story just came back. And it's interesting because you just mentioned, connecting with Graham, who's wonderful. He's been on the podcast. I've met up with him multiple times. He is the nicest scientist, nicest guy ever, but also an encyclopedia of all sorts of. He will remember.
Yeah.
So, and one interesting thing that you just mentioned is that you got in touch, and then you kept in touch, and you never know when you get an opportunity to then, join a lab, but also rely on others and potentially collaborate with them. So I think that's an important thing. Oftentimes in the podcast, we talk at the end about advice to young scientists, and the word networking, the buzzword networking comes up. I hate this word. It's just, it's not you're not going out to network. You're just going to talk to people, and you never know what happens. The magic happens once you get out of that comfort zone.
Yeah, no, absolutely. And it was, completely a sort of serendipitous sort of occurrence. And just by chance that I happened to be going to Glasgow, and it worked out really well. And, as I say, Graham is very much an encyclopedic knowledge. You can't have a conversation where he doesn't know every paper related to everything that you've ever. So yeah, it was an interesting experience when you come as a postdoc, just out of a PhD, and but it's been good. Glasgow's been good to me. So
I'm glad. Did you always know you wanted to be a scientist? I mean, there was the med school aspect of it.
Kind of, yeah. So during my undergraduate, as a typical North American, going into university with very relatively good grades in biological sciences, you're pushed towards medicine. And I spent most of my undergrad expecting to go into medicine, but I'd always expected to do research as well. So when I was applying to medicine, I was looking at doing sort of joint MD, PhD type programs and these sorts of things. And in the end, medicine didn't quite work out for one reason or another. And sort of I ended up in research. And having looked back, I can say without any question, I would have absolutely hated medicine. It would have been the absolute worst place for me to have ended up. Not that, just my personality, it just wouldn't have meshed with sort of the way I operate. But I had always, always wanted to go into science from, as long as I can remember. And always, certainly from the time I was in my undergraduate, I'd always planned to do research in some capacity. But yeah, I did have a period where it was sort of, expect sort of medicine is kind of the obvious sort of place to go.
Yes, I got rejected a couple of times also from med school. And I have to say that every time actually 2 or 3 times, every time it happened, I felt relieved, because by that time I was working in the lab. And I, just similarly to you, I realized that is not who I am.
Yeah, absolutely.
It's a personality thing. So much to the chagrin of my father, who was always hoping, but no, that was not for me. And I think it was a blessing in disguise at the time. So tell me a little bit, please. Go ahead. No, go ahead. I wanted to get a sense of when you were during your postdoc. Did you ever think, oh, I don't want to stay in academia, I might want to go in industry? And how was that transition to the current position you're in?
Yes, so my transition is a little bit of an atypical one. Let's say, it doesn't take the model of going doing a postdoc and then being recruited to a tenure track position. So the UK system is already a little bit different. That when you start on, well, they don't have tenure track positions per se, but they have sort of equivalent type positions. So the way things went, I moved to Glasgow in 2010. When I moved here originally, I was on a Wellcome Trust project grant. So it was a 3-year project grant funded by the Wellcome Trust, and that was fine. But after I was here for a year or so, I applied for a fellowship from the Canadian government, so the CIHR, or Canadian Institutes of Health Research. So I got a postdoctoral fellowship from them to sort of change my direction of research a little bit, and change, to go into a slightly different area from what I originally started on. And then sort of when I was sort of having a bit of success and getting some papers and things, the university sort of recognized that and put me on, they called it a leadership fellowship, which, what that meant, I still am not entirely sure. But it made me sort of semi-independent, not exactly a postdoc, but not really a fully independent researcher. So I sort of had some autonomy to do my own thing, but I was still working under Graham's supervision. And I was kind of in this sort of gray area of not quite being a postdoc and not quite being a true sort of academic. And I was in that area for almost, 4 or 5 years almost in the end. But it was because one of the main areas that when I came to work here, we were working on the fatty acid family of receptors. And we did a lot of sort of more traditional drug discovery. We had a strong collaboration with a medicinal chemist, and that led to some new tool compounds. And ultimately, we managed to convince a venture capitalist to put some money into a spin-out company called Caldan Therapeutics. And I spent 2.5 to 3 years where half of my time was dedicated to working for Caldan Therapeutics, and half of it was spent working for the university in this fellowship role that was kind of a little bit of teaching, a little bit of research, a little bit of everything. And then it would have been 2018, I guess, when sort of my position with Caldan sort of wound down just for the way the funding of the company worked, that I was finally put onto what, in the UK is called a lecturer position, but it's essentially the same as an assistant professor position, but they don't have the same tenure sort of sort of a thing as with North America. So through that time, I was on a long period of time of being on short-term contracts. So when I was on the fellowship, particularly when I was working with Caldan, companies, and small spin-out companies work on short time scales, so they'd have a year of funding, and you'd have a contract for 1 year, and then so it was a long time of not really being completely sure where I was going to end up. And I certainly had, having worked with Caldan and a few other spin-out companies and various things over the years, have considered, moving into sort of industry. But, I think I've always had this sort of romanticized idea of academia that, that you have the freedom, and, and I had always just assumed that was where I was going to end up. And at least to this point, that's that's kind of worked out. But yeah, I've certainly considered, and I've done a reasonable amount of sort of collaboration and various things with industry and sort of companies and things over the years. But at the moment, academia has always sort of been the sort of draw.
Yeah. Thank you for sharing that. I think it's interesting because I have two questions. Number one, how did you, knowing that you have a short-term contract comes with stress, right? Because you don't know what comes afterwards. Although, especially that you have, you're 1 foot here and 1 foot there. So number one, how did you manage that, or how did you deal with that?
I don't know. I kind of always just felt like it was going to work out here. So the situation, in the UK, the universities, well, Glasgow in particular, is a little bit more receptive to taking a postdoc and keeping them on to a permanent, academic post. Whereas I know in North America that tends to be fairly uncommon. And while I was a fellow, I was sort of working closely with the academic staff. I was doing teaching, so I always kind of just, I don't know if it was just blind faith or just sort of felt like I assumed it was going to work out. So there were a few times when I was a little bit stressed about what might happen. In particular, sort of towards the end of the fellowship with Caldan, I applied to other positions, and I applied to a couple of the universities in the UK and at least one university back in Canada, and ended up being offered a few permanent positions at other universities, and that was kind of the sort of, let's say, precipitating force that made the University of Glasgow finally move me to the permanent contract. And sometimes that's the way things go, until you have sort of another offer, things don't really move.
Yes, when demand is higher, and then you showed that you were in demand on that end. That's fantastic. Number two, what was the difference working in small biotech versus in academia? I think this is, if any, on your end, because you were in a very special position in the sense that when you're a full-time employee in biotech, I can speak to that. But when you're an academic and you help in, that might be kind of maybe a different way of going about it.
Yeah. So the structure of the way it worked for me was that I was always employed by the university, and that that Caldan contracted research back to the university that then bought out my time. So I was never an employee per se directly of the company. So it was different. It was a really, really useful experience to be involved in sort of a small scale sort of drug discovery, and the way that Caldan as a company was structured, it was kind of a virtual company in terms of it didn't have its own research offices. It employed CROs to do the majority of the work, and then some of the work was contracted back to the founding universities. So being involved in that and working very closely, we're working with the company Signature Discovery, which is sort of one of the larger CROs in the UK. I guess they're now expanding outside of the UK as well. But so being involved in a project where we're working closely with a company that does integrated drug discovery, and really seeing how drug discovery from an industrial point of view sort of is approached is a bit different. And I think as a company, Caldan, although it was a small spin out, really approached the project more in a traditional drug discovery sort of way, as opposed to some other small spin-out companies that operate out of the university tend to be a little bit more academic in the way they deal with things. Caldan, because it was doing all of the work through Signature, the sort of research was led by people that were big pharma people that were running the program. It was very much a traditional drug discovery approach, even though we were a small company. So seeing that, I think, was a really useful experience, because in academia sometimes you have a bit of a you find some compound that acts, and then you put it in a mouse and it's fine. In obviously you don't know anything about the PK and any of the other details of so learning how important those things are from an industry point of view was really incredibly useful as an experience. It had some downsides, in terms of, ability to publish papers. If 50% of your time was spent on really sort of a true industrial drug discovery program that was not going to lead to publications, it limited how productive I could be in terms of that the sort of academic side of things, and maybe that's caused issues later on. But I think it was a very useful experience. I certainly learned a lot from doing it.
I like that. I like that. I think finally, I'm not the only one saying these things. So I think from a biotech perspective, things are fast. There are steps that you need to take. And to your point about publications, you may or may not be able to publish. And if you publish something that you've concluded and finished up, and it's public knowledge, and it's been a while. That being said, sometimes these things also happen in academia. Just to give you an example, September 20th, we published, so I was author, I think, number 6 out of 100 people to publish a science paper for which I have finished. I conducted and finished the experiments in 2015. So that also can happen, but it was a fantastic opportunity. It was an amazing team, and we put something down that's important and interesting, but also helps patients understand. So we actually characterized germline mutations in GLI2, and that have clinical manifestations in patients. Then again, it took 9 years to get there. I was reviewing my data a couple of weeks ago, 9 years later. So anyway, but it really depends, on what drives you as a person, whether it's, the more academic type research or the more industry. Both can be very rewarding. And I'm glad you had the opportunity to experience a little bit of both.
Yeah, no, it's been interesting.
From a program or research program perspective, once you became a lecturer, how did you go about setting up the topics you wanted to work on? Is it similar to the US, where you get your position, you need to find your own voice in that sense in your own projects, and you need to stay away a little bit from what you've done in your postdoc? So
I think that's an area that I've found a little bit challenging to negotiate. Because I spent this sort of period where I was semi-independent already, and when I was semi-independent, I was working on areas that were close to the areas where Graham worked. So sometimes similar receptors, but maybe looking at them in a different way or different approaches. So yeah, trying to find that niche that carve out what makes you different has sometimes been a little bit of a challenge. And in the UK system, where when you start, you don't get a big startup package to build a lab, you get shared lab space with someone else. And obviously the shared lab space that I had was with Graham, who was also my previous supervisor. It does create some of these sorts of challenges, let's say. So as I've built out, some aspects of it have been trying to build with, using different receptors. So broadly, my research is focused on a family, or it's not exactly a family, but a group of receptors that respond to metabolites. So we're interested in sort of the sort of group of receptors that respond to metabolic intermediates. So historically, I've done a lot of work on fatty acid receptors, but more recently, we've done a lot on the succinate receptor and the HCA receptor. So various receptors that are involved in sort of responding to these sort of metabolites and metabolic intermediates. And whereas Graham has kind of approached most of the stuff that he's done from a drug discovery point of view, I've tried to sort of take a step back a little bit from drug discovery and try to build it, ways to understand sort of more of the physiology of these receptors. So I think having worked on starting with the cannabinoid receptor, but then onto the fatty acid receptors, one of the big questions in these fields has really been, where do the ligands actually come from? How are they actually being activated in a physiological setting? We know that endocannabinoids are there, but they're very lipophilic. Are they actually being released? The same thing with fatty acids. We know that there are very high concentrations of fatty acid in circulation, but most of that is bound to the albumin and not accessible to the receptors. So where is it that the ligands actually come from, and in a physiological sense, how are the receptors regulated? And that's kind of been the goal of sort of what my independent research program has sort of been aiming to work towards. And they're quite big questions to answer. And but that's sort of what we've kind of tried to focus on.
I'll ask this first and then I'll follow up on what you just said, because I always ask this question: what's your favorite GPCR?
My favorite GPCR, I mean, the one that I've done the most with is the FFA4 free fatty acid receptor. More recently, a lot on the succinate receptor. So those are probably the two that we've got. I have sort of, the most of, I don't know, recent interest with. But as I say, that across the group, I've worked with all of the fatty acid receptors quite a bit, and the various other metabolite sensing ones.
It's interesting. I was talking to someone earlier this week. Actually, I was talking to Terry Kinnakin earlier this week or last week, long story. And I was mentioning that whenever I think about a GPCR, especially when something that I've never worked with, you need time to get to know your receptor. And you need time to figure out what are the conditions in which your receptor is going to start speaking to you. Because, they're fascinating, but each GPCR has its own language. So I think from a metabolite sensing GPCR's perspective, they're in a different category. Let's on an island there, because we don't know enough, and it's hard to figure out what activates them and how.
Yeah, and I mean, often we can find out what the endogenous ligand is for them, and that's not too difficult. But where that ligand comes from, and how it actually works in a physiologically or pathophysiological setting is often quite a complicated sort of thing to deal with. And then the other challenge you often have, particularly with metabolite receptors, is that, most of the ligands can be made by any of the cells that you're working with. So if you're working with the succinate receptor, every cell has the ability to make succinate. And whether that succinate gets released or not to activate the receptor is hard to say. But those are sort of the important questions, and they can create big issues, and certainly we've had plenty of issues where you have sort of very high constitutive activity in signaling assays, and you're always unsure, is that because the receptor is actually constitutively active, or is it because I just can't get rid of the natural ligands? And for a while we were for some of the ligands or some of the receptors we use dialyzed FBS to try and remove small molecule and all these sorts of things, and it does work to a degree for many of them, but it is one of the big challenges with this family of receptors. And as I say, when you do that and you have those sorts of challenges to pick out what's going on in, say, a hex cell, and then imagine trying to do that in an animal and really figure out how the signaling pathways work, it really becomes quite challenging.
I can only imagine, because when you think about it, even the media, you just mentioned dialyzing FBS, you don't know you can ask and get the information as to what's in your media, but even then, you don't know if something is in the media. I've recently been in a situation where we had cyclic AMP in the media, and we were trying to measure cyclic AMP. So you couldn't get any dose response effect, because you were saturating your whole system there.
Yeah, I mean, these sorts of things can be a big issue, but again, for some of the succinate receptor, if you have media with high glucose is, usually succinate doesn't get released from the mitochondria, but if the cells experience any sort of hypoxia, then you tend to get succinate being released. And whether your cell culture conditions are experience hypoxia, even the height of the liquid that's above the cells can affect how much oxygen gets to the. So there's all sorts of these things that do create complexities in terms of how you can understand what these sorts of receptors are really doing.
What are some of the approaches that you've been using to interrogate these receptors? Clearly, they're challenging.
Yeah, so I mean, my background was in my PhD was, as I say, was a dimerization sort of project, and we were using sort of the traditional Brett sort of type approach to look at dimerization. I learned to do Brett in Harry Hebert's lab in Montreal. I spent a few weeks working in his lab to learn how to, and I think that that's kind of stuck with me, and I've continued to use sort of Brett and these sorts of biosensor type approaches. So we do a lot of work using the whole range of different biosensor type approaches, and often with the goal of trying to move away from doing that in hex cells and move into doing it in something that's a bit more physiologically relevant. So one of the big areas that we tend to work on, we do a lot of work in adipocytes as one of the main cell models that we use. So we we express sort of biosensors to try and study fatty acids and how fatty acid signaling or how succinate signaling might work in adipocytes. One of the other projects that I've sort of got involved with recently, and this is, you know, sort of a bit of a, you know, the way things work out. We have, so at the moment, the research facility that I'm in, it's called the Advanced Research Center at the University of Glasgow. I'm not sure where the name came from, but it's a very interdisciplinary sort of research center. So it has a mixture of engineers and people that do cell engineering, people that do, you know, so all sorts of different things. And the idea of it stimulating interdisciplinary collaborations, and that's led to us doing a fair bit with a cell engineering group to look at how you can grow adipocytes in 3D culture, and then we can use 3D cultured adipocytes as a more physiological model to study how the receptors signal within that environment. And sort of, you know, this is sort of come about entirely because we've built in this connection with these engineers. More recently, we've also started looking at sort of how the mechanical properties of the adipose might affect the receptor signaling, and we're working with someone that uses sort of atomic force microscopy to look at how the changes in sort of mechanical properties of adipocytes might be affected by the receptor signal. So we've taken quite a broad range of approaches, but you know, the most sort of consistent theme has always been using the sort of classic GPCR biosensor type approaches, whether that's Brett or nanobit complementation, or, you know, all of these types of things.
I like that. I think this is really cool. I think 3D cultures are really cool. Just thinking about them, it's an interesting setting because it allows you to look at something outside of an organism, but still it gives you that dimensionality of the tissue.
Yeah, that's certainly the hope, and obviously, you know, reducing animal usage and all these sorts of things are sort of big themes in research. So that, you know, originally one of the students had a project that it was meant to be a completely animal-free project. So it was sort of, she called it a vegan, you know, project. So it wasn't allowed to use FBS, and quickly she found out that the cells that she had would not grow in any of the FBS alternatives that we tried. So it made it a little bit of a challenge, but yeah, there is lots of these sorts of complexities and things.
How large is your group?
So at the moment, I have 6 PhD students. And then we have various undergraduate and master's students that in the UK system, that you tend to have undergraduates for sort of 3 or 4 months a year, and master's students for 6 months a year, kind of thing. So I at the moment I have 6 PhD students. Yeah.
Do you also teach while doing research?
Yeah. So I do. So as I say, my job title is lecturer, but it's a lecturer in a research and teaching position. So it's a mixture of both. I think it's meant to be 30%, something like that, time teaching, but the time is a bit loosely controlled how much. So I do a reasonable amount of teaching, but what I do, I'm essentially responsible for the final year of biochemistry program for the whole university. So even though I'm more of a pharmacologist than a biochemist, the way the university structure is gone, I sort of run the sort of final year of biochemistry degree program at the University of Glasgow. So some teaching associated with that, but a lot of teaching admin and organizational type stuff. So a bit of everything, as most academics tend to be.
Of course, and then the follow-up question is, how do you balance it all? Because 6 PhD students, you obviously have a life outside of the university and teaching. Yeah. Any advice, any tips and tricks that you've developed over the years?
I don't know. I mean, 6 PhD students is of quite a lot, and you know, in some of it, I'd like to say it was planned, but I sort of went from the way PhD students are funded here, you kind of have to get individual funding, and it can be quite difficult to get PhD students at times. So when I first started as a fellow, it took me sort of 3 or 4 years, and I didn't have any PhD students. And then all of a sudden you get one, and then it somehow, and you ended up with a whole lot all at once. So, and you felt like you kind of need to, when you get the chance to take them, you go for it. So, yeah, in terms of balancing, it can be difficult. Particularly if the teaching load becomes a bit heavy, and there's a lot of the admin. Obviously, this time of year when classes start, and particularly running a final year program, there's sort of a whole new set of students that we need to get sorted out, all their research projects and all these sorts of things. So it does take time. Work-life balance, I think, is always a difficult one for anyone in academia. It's always a real challenge to figure out how to do that. I'd like to say when I was a postdoc, I had a work-life balance, but I worked very long hours as a postdoc. Less so now, but there are still times when you have deadlines coming up, or where you're trying to get a paper finished, where it's unavoidable that you're working these long hours. But it's nice when
it's good.
It's nice when you're working on those sorts of things to finish a paper or something that where it's things that you're excited about. Whereas working long hours for more things that are less exciting can sometimes be a little bit more of a struggle, let's say.
I know, doing radioligand binding assays, and having 120 files that you finally at 6 p.m. need to take down to the counter. And then you come back the next morning, and after the third tube, the counter got stuck.
I've had that experience. When I first started, we had a fairly old counter, and it had a dot matrix printer attached to it. So it didn't save the files. It just printed off. And I put in this massive experiment with, it was a huge amount and I think it took 2 or 3 days to read, and we left it over the weekend, and the printer jammed and just printed all the values on top of each other. So it had to be read out. Yeah. So yeah, I've had that.
It was not scripted. Anyone listening, I didn't know about your experience. I was simply explaining mine, or the paper, or the printer runs out of paper, or things like that happening. No, I do agree. I think from a busyness and perspective, I think we're all busy. So in addition to running DrGPCR with a fantastic team, so I show up to podcasts, basically. We got to a point. I also work at a biotech company, and it's extremely busy. So the pace is fast. So there's a couple of things that I found that help me keep my cool, having a to-do list. The minute I don't know what I have to do, I'm like, okay, I have too many things. Just keeping the to-do list and prioritizing it has been very helpful. Tracking my time also has been something very helpful, where I know I can say, guys, I've been in too many meetings, now I need to focus. Or protecting my time, which is a challenge because you don't want to say no. There's always something to be discussed, but at the same time, there's data that needs you to look at it. So tough.
No, I think it is one of the it's a common thing among a lot of academics is you have the tendency to not say no to things, and I am most definitely guilty of that at times, and where you just say yes to everything, and then you end up being a bit overloaded.
Exactly. I started declining meetings. Or also, for example, yesterday I was supposed to be in a meeting, and I was looking at it, and I was like, why am I in this meeting? I don't need to be here. So I basically just, gave it off to a colleague and said, you take care of it. I don't need to be there. And it's a tough balance to get to.
Absolutely.
All right. So then I'm looking at the time. Let's go and inspire young scientists a little bit. Top 3 aha moments that shaped your trajectory.
Top 3 aha. So I think that the first moment the first moment that got me excited about research was actually my very first experience in a research lab. So I did my undergraduate in a small university. It wasn't a particularly big science university, but in Canada you can get these Natural Science and Engineering Research Council summer student awards. So as a second year undergraduate, I got an award to work in a research lab for the summer. So I turned up, and a second year undergraduate student, little experience. And we come in, and there was one other woman working in the lab, and she had at least worked the summer before, so knew a little bit about what was going on. And we came in, and the sort of PI gave us an introduction and said, all right, so we have this research project where we have this toxic fungus that grows in water damage, and it's apparently linked to these awful health effects. What I need you to do is come up with a way to see what the lowest dose of this fungus we can give to a mouse where it has an adverse effect on their lungs. And then he left. And that was sort of it. And he left it to the two of us, so this, to sort of come up with a plan for how we would do it. And, I know a lot of people would be very intimidated by, that kind of situation. But actually, at least for me, in my personality, I found that, it was just an exciting thing to try and, come up with what we were going to do. And in the end, we said, we decided to measure, the concentration of cytokines in the BAL fluid that we washed out of the lungs, and which was fine. But again, it was a small university, so we didn't have a plate reader to measure. So the first week we said, we need to buy this, $15,000 plate reader. And he's like, oh, fine. Well, so it was an odd experience. Let's say, now thinking if I would ever consider doing anything like that to a second-year undergraduate, I would say absolutely no way. But in our case, it worked out. We ended up getting, I ended up getting 2 papers out of my undergraduate work. It was, the project worked out. So it was kind of as I say, it got me very excited about doing research. And then, I guess a second one, probably similar, was in my PhD when I started, my supervisor just sort of handed me a grant and said, read this and see what you're interested in and come up with, what project you want to do. So, I think that was again, it's the common theme of sort of the freedom to sort of, choose and to design, where the research goes.
I love it. And let's circle back to your PhD when you were given the grant and you picked GPCRs. What got you interested? What piqued your interest?
I don't know. I mean, I've always liked the molecular stuff, and even I remember back to my undergrad, going to lectures and hearing it. I actually really remember the lecture where they went through GPCRs and having lots of questions even back then about how they work, and does one receptor activate multiple G proteins and all these sorts of things, and the person who was giving the lecture probably didn't have answers to most of the questions that I probably had at the time. So it's always been something that, as I say, I didn't actively choose to go to a GPCR lab, but then what they were doing, that was what I was most interested in, and then you sort of get sucked into it. Maybe they say, after a few GPCR retreats, there's no turning back. No,
I agree, and it's interesting because I also got to working in a lab during my second year as an undergraduate, and I've told this story many times. So for those listening to the podcast regularly, I apologize, but I wanted to work with Michel because Michel Bouvier was teaching GPCRs, and one thing that fascinated me at the time is you have heterotrimeric G proteins and you have different G alpha, beta, and gamma subunits, but how do you know which combination works best with what receptor? And so I ended up not working in Michel's lab for the summer, but in working with Nicolas Eveker, who he was just hired, and then that's where it started. And actually the work I started during my undergrad ended up being my master's project, and I got a paper out of that. And I was hooked the first hour, thinking, ooh, you have all these proteins doing all sorts of really interesting things, and how do they know what to do and when?
Well, it is a big question that we probably still don't really know the answers to.
Exactly. There's much more. Any other aha moments?
Any other aha moments? I don't know. I'm not sure the time that I've obviously, I mean, certainly in terms of the moments that have shaped my career, moving to Glasgow and starting here was obviously the biggest thing. But I can't think of any particular sort of single moments or single findings that, like I said, something happened and has changed sort of my perception of things. So I don't know.
That I think one aha moment is we'll take that. We don't have to go to 3, but any advice for junior scientists, early career scientists who want to contribute to the field? Anything you can think of doing differently, for example, or
I mean, in terms of advice, I think the biggest thing is having something that you're excited about, being excited about what you're working on is really just so important that it makes you want to, read the literature. It makes you want to, come up with the next experiment. And that really is, the biggest thing. If you really are, excited and want to see what happens next, that it makes it so much better coming to lab and capping those 200 vials that you have to then and if you're excited to see what comes off the counter the next day, then it makes it a little bit more manageable to do those sorts of things. So yeah, I mean, that's kind of, I think, the biggest thing. I mean, you can say things, making sure you network and you build out your connections and get to know people and go to meetings, talk to people, and see what opportunities come up. And you never know where the opportunity is going to come from. I mean, let's say I met Graham through a random chance, and somehow I've ended up being in Glasgow for 14 years, and now basically here for the long term. And it was not planned. It's just the people you happen to meet. So talk to people at conferences and see where it goes.
I love it. I know you have 6 PhD students and a lot of undergraduates and master's students coming and going, but if and when you have job openings, where can people find you?
I mean, it would all be on the University of Glasgow. I've got my website on the University of Glasgow. We post things on LinkedIn if we have openings coming up and all of those sorts of things. So, I think that's the main places.
Send us the ads, because we do have a space in the ecosystem as well to post these job ads. More than happy to do that. Last question. I think this is the toughest question. What should we title this episode? I think one theme that came out from our conversation is the odds also from making it from Halifax to Glasgow. I think are also, when you go back and you remember those times when you started an undergrad.
It was definitely not where I expected to be, that's for sure. I very much assumed I would, do a PhD and then go do a postdoc somewhere in the States and then come back to Canada and, start a lab. That was always what I just assumed I would,
Yeah.
So, well, you never know where things will take you, I guess.
Well, there you go. You never know where your GPCR takes you. When you think about it, one is the freedom of being creative and working on topics that interest you. That's a theme that came through our conversation. But it was your work on the cannabinoids and being in the GPCR lab that allowed you to meet Graham, keep in touch, and then take you from Halifax to Glasgow. And then it's still the GPCR the GPCRs that are keeping you in science and also where you are today.
Yep.
There you go. There you go. We actually left our very factual world and we came up with a hopefully inspiring title. We'll see what people are going to say about that.
We'll see. We'll see. It's always the hardest thing. It's even from when you're writing a paper, the coming up with a title for the paper is always the hardest thing.
I think so too. And then you come up with different versions and it doesn't sound good and it doesn't say much or it says way too much. You want people to actually read it. So tough one.
It's always a struggle.
Absolutely. Brian, thank you so much for your time. Don't go anywhere, but it was a real pleasure chatting with you.
Thanks. It's been great.
I need a favor. If you enjoyed this episode, please click the like button. And if you like the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribers yet. One click goes a long way. All the information about DrGPCR is in the description. Find all our fantastic features and everything you can benefit from becoming a premium member. And while you're on it, check the DrGPCR University courses and share them with your network, colleagues, and friends. We also want to hear from you. Is there anything missing from our ecosystem? Can we make it better? Email yamina@drgpcr.org or email our team at hello@drgpcr.org. Thank you so much for joining us in this podcast episode. If you're still there, we also want to take a minute to thank our team members. Yamina, Attila, Montserrat, Cam, John, Yatsu, Mark, Ivana, and Andreina. If you like our podcast, you can leave us a 5-star review on Apple Podcast. Until next time, and stay safe.